During embryonic development, a subset of cellular mRNAs is trafficked and locally translated within axons. We are interested in understanding the role of this local translation in neurodevelopment. My laboratory has found that local translation is important for the synthesis of several cytoskeletal regulatory proteins, such as RhoA and PAR3, and that the translation of these proteins within axons is important for mediating the effects of axonal guidance cues. However, we have found that axons contain transcripts that encode other types of proteins. For example, we have found that axons contain transcripts encoding transcription factors, such as CREB. We were very intrigued by a paper by Van Wang's group at Duke University, which focused on the specification of neurons in the trigeminal ganglia. They found that during development, axons from the trigeminal ganglia encounter BMP4 in the face, which results in the generation of a retrograde signal that is important for specifying neuronal identity in the trigeminal ganglia. BMP4 mediates its effects by inducing the phosphorylation of SMAD transcription factors. During the course of their study, they found that SMADs are found not only in neuronal nuclei, but also in the axons of trigeminal neurons. This raised several questions. How did these transcription factors get into axons? And what is the role of these axonally localized transcription factors? These questions prompted us to initiate this study. Now I would like to introduce Dr. Sheng-Jian Ji to describe the major findings of our study.
The embryonic trigeminal ganglion is a readily accessible system to study neuronal specification and retrograde signal. The trigeminal ganglion conveys sensory information from the face to somatosensory centers in the brain. The trigeminal ganglion has three main peripheral axonal branches, the ophthalmic, maxillary, and mandibular, which innervates the corresponding regions of the face. Sensory information is then conveyed from the ganglia to the brainstem nuclei through a centrally projecting axon bundle. The neurons that innervate each of these regions in the face are spatially segregated into specific domains within the ganglia and exhibit distinct gene expression profiles. In this study, to culture the trigeminal ganglion neurons and to study the retrograde signaling, we extensively used this device called microfluidic chambers. The chamber is mounted on the cover slip and generates two compartments which are connected by narrow microgrooves. The dissociated trigeminal neurons were plated in one compartment, and after 2 to 3 days, the axons grew into the other compartment. We studied retrograde signaling by applying BMP4 to the axons, and then monitoring the accumulation of phosphosmad in the nucleus. We found that co-application of protein synthesis inhibitors to the axons together with BMP4 blocked retrograde signaling, which told us that BMP4 retrograde signaling requires local protein synthesis in axons. Using different approaches, we found that SMAD proteins and the messenger RNAs encoding them are localized to trigeminal axons. By using a special RNI technique, we knock down SMAD messenger RNA selectively in axons. Using this technique, we show that axonal translation of SMAD is required for BMP4 retrograde signaling. We wondered what regulated the local synthesis of SMAD in axons. We found that BDNF is the most efficient factor to induce local SMAD translation in axons. By analyzing BDNF knockout mice, we found that BDNF is required for SMAD synthesis in axons and is required for neuronal patterning in the trigeminal ganglion. Based on these results, we have the following model. BDNF and BMP4 are expressed in the ophthalmic and maxillary, but not mandibular regions of the face. When axons encounter BDNF, SMAD synthesis is induced. If they also encounter BMP4, signaling endosomes containing active BMP4 receptors are trafficked to the nucleus. These endosomes traffic together with axonal SMADs and keep them in an active phosphorylated form. In the nucleus, phosphosmad triggers gene expression pathways needed for neuronal patterning and for the guidance of centrally projecting axon bundle.